So today we're going to talk about what happens when there are mistakes in meiosis. So we've talked about crossing over and we've talked about sorting the chromosomes independently to cause all kinds of different variation in the genetic material that we get in meiosis. Now what we're going to do is we're going to talk about what happens if there are some mistakes when you go into that process of sorting out the chromosomes. What this is called is non-disjunction. So in non-disjunction, these mistakes happen, and it can be that there are chromosome abnormalities. It could be that there is a missing chromosome. It could be that there is an extra chromosome. Or it could be that there is an irregular chromosome. So chromosome that has two of the same kind of part, that it duplicated twice, or there is a part that got deleted. So it's just something's wrong with that chromosome. Some of these chromosomal abnormalities are caused by the chromosomes breaking during cell division. They could get cut when the cell is being pulled apart. But most of this is being caused by non-disjunction, which is a special kind of abnormality that we're going to talk about today. Okay, this is the failure for the chromosomes to separate during meiosis. So instead of pulling apart, they stay stuck together. And so instead of having a cell with each with its own set of chromosomes, we have one with twice as many chromosomes. This usually happens when the chromosomes don't separate in meiosis 1, but it can also happen in meiosis 2 when the sister chromatids are supposed to separate. In this example that's in the picture on the slides, this is an example of non-disjunction during meiosis 1, and so the homologous chromosomes are not separating, and so you end up with a cell that doesn't have any DNA in it at all. In the end of non-disjunction, we end up with 4 abnormal daughter cells. Okay. We have 2 daughter cells that have an extra chromosome, and we have 2 daughter cells that are missing a chromosome. Now remember, in this example, in the picture, this is only showing 1 chromosome homologous pair. In humans, we have 23 of these pairs. So this is a very simplified picture because We can't draw all 23 pairs in the picture and have us be able to actually see it and make sense. So when it says that the picture on the right-hand side is missing a chromosome, it still has the other 22 pairs. It's only missing the one that is in the extra on the left-hand side. All right. So I would like you to take this picture and explain what I just talked about. I want you to pause the video here and write down what is happening in this diagram. Remember that humans have 23 pairs of chromosomes. This is only showing one pair. Most of these chromosomal abnormalities are really severe and will cause cell death. Okay, so most of these, the cell doesn't even survive enough so that it can be used for reproduction. Most of the cells will die before they get to that point. But sometimes these do not cause cell death, and they can cause problems in an offspring because the gamete with the chromosomal abnormality is then passing on that abnormality to the offspring. Now, just because an individual has a chromosomal abnormality does not mean that they are an abnormal person or that they are at an extreme disadvantage. It just means that their genetic code works a little differently than everybody else's, and they might have a different set challenges than everybody else does. Doesn't mean that there is anything bad about them. They are generally very good people. Okay, so we're going to talk about that a little bit in just a second. One of the most common types of human chromosome abnormality is called Down syndrome. Down syndrome is an abnormality where there is non-disjunction of chromosome number 21. If you were to go back and look at the karyotype drawings that we did, number 21, an individual has 3 copies of chromosome 21 instead of having just the 2. So they get an extra copy of that chromosome from one of their parents' gametes. Individuals with Down syndrome, they can have intellectual disabilities, but they can also not. They usually have stunted growth, and they have certain facial features. It is also called trisomy 31, meaning that there are 3 copies of the chromosome 21. An offspring inherits their Down syndrome when one gamete with an extra 21 chromosome fuses with a normal gamete. All right, and so this is an explanation of that. So gametes are only supposed to have one copy of each chromosome. The gamete with the extra chromosome 21 fuses with the normal gamete during sexual reproduction, and then it causes there to be three copies. The cell divides over and over and over again, and so every single copy of every single cell in that organism has 3 copies of the 21st chromosome. And we can talk more about different ways that different disorders can get passed on when we talk more about genetics and heredity and the passing on of traits when we go into this topic more in April. So there are a couple other genetic disorders that are caused by chromosome abnormalities. If humans have an extra copy of chromosome 13, it causes Patau syndrome. If you have an extra copy of chromosome 18, it causes a disorder called Edward syndrome. An extra X chromosome in males causes a disorder called Klinefelter syndrome. And if there's a missing X chromosome in females, it causes a disorder called Turner syndrome. And we're going to talk more about those and a bunch of other different genetic disorders when we are finishing up our genetics unit, because we're going to do some genetic research projects. All right, so those are our notes on non-disjunction. You are going to have an Edpuzzle to do today, and just make sure you get your notes and you fill out and complete the question where you are supposed to explain what is happening in this picture. I am going to be checking this in particular in your notes on Wednesday. All right, I will see you Wednesday. Have a wonderful day.